All right, welcome. All right, so welcome to the Morgan State University Patuxent Environmental and Aquatic Research Lab, or the PEARLS Summer Internship Symposium. We're so excited to have you here. I did mention in my email that masks are required. They're actually not. So if you'd like to continue wearing your mask, that's great. But if not, then you're welcome to take it off. We have a wonderful group of 5 interns this year. I'm so excited for them to give their presentations this year. But I did want to acknowledge our mentors before we get going. So we have our econ team, Dr. Scott Kanaki and Caitlin Ritchie, who is bringing in the stragglers. And then we've got our aquaculture team, as well as Dr. Chun-Lei Fans. We've got Dr. Ming Liu, Brittany Wolf-Bryant. So we're so excited to have them as mentors this year. If we didn't have them, we wouldn't have an internship program. If y'all were able to grab the schedule in the back, if not, you're welcome to go grab one. So we've got 6 talks. One of our interns decided to do 2 projects, so she'll be presenting twice. But we do have a short break in the middle of it, partially to catch up on time, partially to give y'all a chance to stretch. We are on Zoom. Like you saw, I was checking the sound there, so we will be taking questions on Zoom. This is also recorded. So if you'd like the recordings afterwards, please email me or check our YouTube page. It'll be as one long recording as well as broken up into talks. And I think, and just for y'all mostly, watch out for all the cords. I could not make them neater than that. So with that, I'm going to ask Brittany Wolf-Bryant to come up and introduce our first talk.
Hi, everyone. Thanks for coming. So I'm happy to introduce Maya as our first intern presentator. She is a recently graduated from Morgan State University. We're really happy to have her this summer. She did such a great job. She took up an interest in filming a lot of the things that we're doing. So if you are interested in some of the things that she did, check out her YouTube page as far as Maya can see. So I will let her talk about some of the research that she did this summer. So come on up.
Thank you, Brittany. That's me. Good morning, everyone. My name is Maya Sharp, and I'm a recent graduate of Morgan State University. And some of my project is on using PCR assay to detect dermal disease in Maryland waterways and eastern oysters. So oysters are significant for many reasons. They create a source of food for humans and wildlife. They construct habitats for hundreds of small and juvenile fish, crabs, mussels, shrimp, and many other species. They have economic value as well. So according to NOAA's fisheries, the restoration of oysters can bring in a 150% increase in blue crab harvest and an additional 10 million in annual fishing revenue. And finally, they improve our water's quality. So the average adult oyster can filter up to 50 gallons of water a day. So prior to the dramatic decline in our bay, the oysters were able to filter out the entire bay, which is 19 trillion gallons in just 1 week. However, unfortunately, with the remaining oysters that we have now, it takes them about a year to do so. So the oyster populations face a variety of threats: habitat degradation, predation, mass harvesting, climate change, and disease. So the spread of disease and climate change are directly related. So since dermal was first detected in the Gulf of Mexico in the 1940s, it is used to those warmer waters. So now with climate change and the increasing temperatures spreading up the East Coast, the disease has been able to spread and make its way up as far as the New England states. And it also increases the winter temperature, so it widens the window of opportunity that the disease has to infect oysters. So there's many diseases that infect oysters, but the one that remains top offender is the endoparasite Patuxent marinus, also known as Dermal, that is responsible for mass mortalities experienced by the eastern oyster. So the way the disease is transmitted, you have an uninfected oyster, and then ingested, then it makes its way through the gills and the mantle, and then it proliferates throughout the oyster, making its way to digestive glands, and then ultimately causing tissue lysis, enters the bloodstream, causing circulation blockage, and then kills the oyster. So this disease does require the oyster to be able to reproduce. The purpose of our study. So despite the dermal prevalency, it has been reported to decline in recent years, and it remains detected in oyster populations in the Delaware Bay. And this project will help us to assess if the mortality in our oysters are caused by disease infection. So this summer we had a task of bag splitting, where we would go to the pier and retrieve the oyster bags, separate the oysters that are alive from the ones that are deceased. And during that, we noticed that significant mortality was in our oysters. So now the question is this caused by disease, or is there another factor in play? So our objective is to investigate if there's any dermal infection in Maryland oysters, and to determine if there's any prevalency difference among different oyster lines. On to the materials and methods. We'll start off with our study region. We have the Chesapeake Bay and then the Delaware Bay. So we have 12 oysters from the Patuxent River lines C and B. The B just means a diploid line, and D is a triploid line. Then we have 10 oysters from line B in New Jersey, 12 from both the Potomac River and Eastern Shore, and they're both commercial lines, for a total of 46 oysters. So first, we shuck open the oysters, we take gill samples, and we store them in 95% ethanol. From there, this is a video of us doing the DNA extraction process. So after we take the gills, we extract the DNA from the other parts, other macromolecules that are composing the tissue, which is the proteins and the lipids. Following the DNA extraction, we must make a reaction mixture, and it is composed with these reagents here. So PCR buffer, DNTP, forward and reverse primer, TAC, polymerase, and H2O. So when we get our DNA extract and our reaction mixture, we mix it together, brings us to our next step, which is PCR, polymerase chain reaction. And this is a technique that is used to make multiple copies of a segment of DNA fragment, resulting in many copies from just a small initial sample. So this is the way the PCR would work. We would take this solution now, enter it into the machine, and it will begin with double-stranded DNA. Now, with the right temperature, the DNA begins to unwind, beginning the DNA chain. So now I have a question for the audience. So by a show of hands, when I get this DNA extract, who thinks that it's just oyster DNA and that I will find? Who thinks, okay. Who thinks I'll find other types of DNA as well? Okay, you're correct. So since dermal is a parasite, it does have its own DNA. So we need to be able to differentiate the DNA from each other. And we do that by using a primer, which is going to be a annealing stage. A primer is just a short nucleotide sequence that's complementary to the DNA of interest, and that's the dermal DNA. So the primer then attached, acting as a template for DNA synthesis, and then with the right temperature, 72 degrees Celsius, and the other reagent in the reaction mixture assists with the actual synthesis, bringing to the extended stage. And this process takes about 2 hours. Following that, we use a traditional method, gel electrophoresis. Now, this process allows us to separate DNA from each other based on their molecular size. So first, we take the DNA, we mix it with a DNA stain, we insert it, like I am right there, into the pores of the gel, and with the electrical charge in the apparatus, it pulls the DNA towards the positive end of the apparatus. So that's how it separates. Now, following the run of the gel, it takes about 40 minutes to an hour. We look at it under a blue light or UV light, and this is kind of what it's supposed to look like. Now, our results. So for the DNA extraction, we tried 2 different methods. We tried the Kellex method, which is a shorter protocol, inexpensive, and you can do unlimited sampling with it. And then the kit method is a longer protocol, more expensive, and limited samples. And you'd be surprised on which one that we picked. So we took the DNA extract prior to the PCR product just to see the quality of the DNA as it ran through the gel. And we noticed that the quality was way better in the kit, so we decided to move along with this method. So after running many gels, we weren't getting the desired results that we wanted to see. So this is an example right here with this gel. we had 3 samples from Eastern Shore, a positive control and 4 negative controls. The problem here with this gel is that you see a strand with every single slot. We're supposed to see one with positive control, of course, but then none with negative control. So we realized that there's a problem that we're seeing in our gel. So we noticed that it is a primer dimer interruption. So primer dimer is just a byproduct of the PCR process. When instead of binding to the DNA of interest, they just bind to each other, like this one right here. And then that's the result that we're going to constantly get. And the other problem with that as well is that the primer dimer PCR product has a 50 to 100 base pair, while the dermal has 85 base pairs. So it's hard to differentiate through the gel if it's dermal existing in there or not. So then we had to play detective a little bit to try to find solutions to exclude the primer dimer. So first, based on what I've seen other researchers do to exclude primer dimer, they increase the annealing temperature. We did that, that did not work. And then my mentor, Dr. Wu, he was like, maybe we should revise the primer itself. So we were going based off a literature, and this was the primer right here with the guanine adenosine right after each other. And then the source that he saw, you see the adenosine guanine. It's a very slight change, but very important. And then when we got this new primer, what did we get? We get it 2 days ago, and then we ran the gel yesterday. So this is very new results. It did work. There was a mistake in the literature with the primer. So we have our positive control band. They are light, because this is an older positive control, but we don't see any bands with negative control, and that's what we want to see. So although we weren't able to detect dermal in any of our samples this summer, through our efforts, we were able to determine the proper primer sequence to exclude the interference of the primer dimer. So for our future endeavors, we'll continue this project using the new primer. You would get a new positive control so we can have more definition through the gel and investigate the prevalence of dermal at other hatcheries and farms in the Chesapeake Bay watershed as well. These are my references. Does anybody have any questions? Oh. As far as Maya can see.
Oh, there it is.
For those of you on Zoom, you're welcome to either speak up now or type it in the chat and I will read it out.
If there's no questions, I
Yes, Nate Cantor asks, can these methods to check for diseases be applied to other bivalves like clams or mussels or specific to oysters?
I believe it can be used for other diseases as well. Yes, I got a nod from my mentor, so that's correct.
So this is a general question, Maya, or kind of an observation / question. When you started this internship, you never worked in a hatchery, correct? You wanted to learn about it, see what goes on in a hatchery. Kind of uncertain whether it was going to be for you.
Yeah.
That's totally fine if it's not after all is said and done. But what's your kind of like after the fact assessments, like everything you worked on, like where are you at on like this type of work, pros and cons or whatever?
Okay, so the back is not working.
What?
Can you press the back,
No,
please?
it's not. Okay, right now. No. That's not. Where do you want to go?
I want to wait, go forward. And then forward again to the question slide.
Yeah.
Okay, now this is a clip of us in the hatchery in one of our more busy days. I can honestly say that the first week, my lower back was hurting with a lot of bending down and doing things. But overall, throughout the experience, I think I learned more here than I've done in any other internship with all the hands-on work. And it being a small, tight-knit office, I've been able to learn from everybody there with my great mentors as well. And what's fulfilling about working in a hatchery is that you get to grow organisms. Like you begin with spawning, and then you're able to grow them and just see their growth. And yesterday, Kayla was able to put one of our larvae cultures into the Mako Cove after it settled on the shelf. So we've seen it through its few life stages. So it was pretty fun. I don't know if I will continue in the aquaculture, but I learned a lot from this experience.
One last question. Do you think that this technique may be used in ED and A capacities, perhaps inside hatcheries to detect diseases / parasites?
It's okay if you don't know. I would believe so, because Genotypic allows you to separate DNA by its molecular size. So I guess once you know the base pairs of the DNA that you're looking for, and then measure it with like a DNA ladder, and you should be able, Dr. Lou? Okay.
Yeah. Come back up.
So moving on with our second aquaculture intern. So next is going to be Jessica talking about her project. She is a rising senior at UMBC, and we were really happy to have her over the summer. She did a lot of great work. So yeah.
So hi everyone, my name is Jessica Bainiak, and as Brittany said, I'm going to be a rising senior at UMBC this year. And my project, well, my first project is going to be on the peaceful sleep, more efficient ways to sedate oysters for sampling. So for anyone that doesn't know, anesthesia is the temporary loss of sensation that can cause relief from pain, muscle relaxation, and/or unconsciousness. So the picture on your left is an oyster that is anesthetized. And so it's a little bit open compared to the tight knitting that you normally see. So for oysters, it was found that magnesium chloride has been proven to be one of the most effective chemicals for oyster anesthesia with the lowest mortality. So some benefits of oyster anesthesia is that usually to get samples from oysters, you would have to shuck them, which in the process kills them. Which means, yes, you will get data from that particular oyster, but you cannot work with that oyster from that point on. So that kind of defeats the purpose if you want to do certain things like sex determination, tissue biopsy, and physiological and genetic studies. So this anesthesia allows you a way to continue work with these oysters from then on. And it's also helpful for the pearl industry that it simplifies surgical techniques used in pearl production and extraction. So the reason that we're focusing on this study is that the chemical that we use, magnesium chloride, is pretty expensive. So the paper that I based my study off of, Sequet et al. 2009, found that the ideal concentration of magnesium chloride for optimal anesthesia was 50 grams per liter over a 16-hour duration. The magnesium chloride, the bottle that you see on your left, that costs $61.20. And at this concentration, every liter of water we would use to anesthetize the oysters in would cost $3.06. So if our setup that I'm about to show, if we had used that concentration for all three, it would cost close to $300, which isn't practical if you're trying to sample a lot of oysters overall. And furthermore, at high concentrations, magnesium chloride is harder to dissolve and creates health risks due to excess heat and irritation of the respiratory tract. So our objective was to create a more efficient, effective method of utilizing magnesium chloride to knock out oysters in a cheaper, faster, and safer way. So here is the actual setup that I used for a couple of the trials. As you can see, there are 3 tanks that each hold 30 liters each, and in them we have 30 oysters, and we would change the concentration of these tanks throughout. So the general procedure of this project would be that we place all the oysters in the tank, and every hour we would check the amount that were anesthetized and we would remove them. We would place a nail to prop them open so we could take a mantle sample. It's a little bit hard to see, but around here, that is the mantle, and there's a little portion of it that has been removed by me. So what I'm doing in this image right here is actually taking a knife and removing part of the sample, because in normal laboratory experiments, they would be using this for sampling. So we wanted to see their mortality rate with normal practices of the lab. So we were trying to replicate these results. So after we removed the sample, we would weigh the oyster and measure its height, because we wanted to see if certain sizes were anesthetized faster or slower than others. And then finally, we would put them in a tank to recover, and after a couple of days, record the recovery rate of the oysters in this experiment. So we ran 4 trials overall. So the first trial, our main thing we were trying to focus on was that a pump. If a pump that filters the water, could we lower the concentration of magnesium chloride and create a more effective way at anesthetizing the oysters? So our first trial was to have the 3 tanks in that setup you saw before at 5 grams per liter. So we were trying 10% and trying to get as low as possible to see what we could achieve with the pump. Our second trial was starving the oysters with the same conditions from the first one to see if starving them would create a higher percentage of anesthesia. So starving them increases the filtration rate. So this might allow more uptake of the chemical, which might anesthetize them more. Trial 3 was our control based off the paper that I mentioned earlier of that 50 grams per liter in the tank. So yes, the 50 grams per liter. And trial 4 was putting a pump in the 50 grams per liter. And I'll explain why I did that later. So for our results, here are the 4 trials, and the paper is the one on your right. So these are the amounts that we should be comparing to and seeing. So this is the 50 grams per liter and the high concentration. Trial 1, as you can see, 3 hours here, it was at around 65% compared to the 80% of the trial, which is not a lot lower compared to having 10% of the magnesium chloride. And trial 2 with starving almost got it up to that 80% that was in trial 3. And both trial 1 and 2 caused 100% of oysters to be anesthetized after that 24 hours. So both of those, I think, were a success and look very promising. However, where the experiment went a little bit off was trial 3 should have been the control. So it should have the exact same results as the paper, but this did not happen in our case. And so trial 4, I tried to put a pump to see if that made a difference. And yes, there were more oysters that were anesthetized. However, in the next chart, the recovery percentage of the oysters, so the ones that actually lived after a couple of days, trial 4 had none that survived. So yes, it might have opened with the pump, but that might have just meant they died in the process. So trial 3, there was a little bit recovered, but not nearly that 100% that we should have been seeing in the other trials. So what we think, so it's kind of hard to see, but this water should be clear. This is what the 50 grams per liter concentration is, and the water should be clear. So we think that our problem of mixing the 50 grams per liter into the water, we could have done different things in actually mixing it. So some potential fixes that people recommended was to pour the water into the magnesium chloride and mix it throughout, or heat it. There were many different things we could have done to try to fix this problem for a later study. So our final thing was the weight and height. We found an overall trend of the larger the oysters were, the faster it actually took them to be anesthetized. So the stars are the ones that were significantly different. While they're not on everything, there still is a general trend of from that 0 to 3 hours to the 3 to 24 hours that after they were anesthetized, it did decrease in the height and weight. So some conclusions of the best protocol to use was definitely adding a pump made a difference, that even at 10% of the concentration, they still had results that were very similar to the 50 grams per liter. I would much rather spend $30 instead of $300. So I think this is worth exploring more. And also starving the oysters from trial 1 to 2, this did create an increase where it almost matched the paper's results completely. So some further steps that we could do is to repeat the experiment at a lower volume of water. We could increase the concentration of the magnesium chloride in intervals to find the lowest amount we can go, or find the optimal amount that we want. We can test the type of pump or filtration system, and we can do more trials related to starving. We could also determine the ideal number of water volume to minimize the excess water use and just really make this a very good protocol to use from now on. So here are my citations. And here are the people I would like to thank. Thank you, Dr. Liu, Brittany Wolf-Bryant, who Lee's also there, and all my fellow interns, and Dr. Amanda Knobloch for all your help, and all the oysters that I killed when I didn't mean to. I'm sorry. Do you have any questions?
Yes, I have a question. How do you know when the oyster is anesthetized? How can you tell?
How do I know when the oyster is anesthetized? Is that correct?
That's correct.
Okay, so it doesn't work. So that the muscle relaxation actually opens the oyster a little bit. Like usually when you take it out of the water, they're closed very shut and you have to shut them to open them. But when they were a little bit open, that you could actually get things in and I could take samples from them. Okay.
We have a question from Zoom. Upon seeing that trial 3 didn't correspond with the paper's findings, did you see if any other researchers had similar problems? Had anyone else in the field struggled to replicate the original trial's results?
That is definitely, we ran out of time currently, but I could definitely go into that more later on. And if I wanted to run this trial and find the best results.
So really nice presentation and well given. I apologize, I missed the first couple minutes, but I may miss this. Was trial 3 run at the same time as trials 1 and 2? Were they simultaneous?
So that was actually a good point. We wanted to save as much magnesium chloride as we could. So trial 1 and 2, all 3 of the tanks were running at the same time. And so trial 1 happened on one day, trial 2 happened the next day. Trial 3, we used one container, one of the tanks, because we didn't want to do all 3 at that 50 grams per liter to save some money. And so we ran those over 2 days. So we did 2 replicates for that. And for trial 4, we did 2 replicates for that.
So could there be any factors doing them in that order that could have caused the problem?
Absolutely.
Yeah.
That there's always variability when you're doing things and if it's not all the same. So some of the factors were at sometimes the oysters might have been kept out longer than possible because we did get a second batch of oysters later on in the week because we decided to run more trials on it. So they're different oysters, there's different timing to it, and we don't know if the conditions in the pier where we took them out might have been different. So there's a lot of things up.
Thank you.
Hi, can you hear me? I don't know if I can ask a question out loud on Zoom. No, I can't.
Someone says something.
Yeah.
Anybody on Zoom have a question?
I was going to try to ask a question out loud, but I don't know if you can hear me well enough.
Yes, we can.
Okay, I was just wondering if you had a proposal or an idea of why the larger oysters were anesthetized faster than the smaller oysters.
So that is actually one of the most interesting parts of this study. In the paper that we were reading, we found that a higher percentage of the smaller oysters were actually anesthetized compared to the larger. So this does somewhat contradict the paper. So there's definitely more research in that. And that was something we did not plan to see at all. So that could also require further research.
Thank you.
Okay. I'll see.
All right, so I'll introduce Kayla since Tamika hasn't gotten here yet. So I'm excited to announce Kayla Mcveigh. She is a rising junior at York College, Pennsylvania, and she worked with Dr. Chun-Lei Fan, Ming Lu, Tamika, and Brittany this summer.
Okay, hi, my name is Kayla. So I worked with Dr. Fan this summer on the evaluation of larval survival and settlement rates in the low salinity tolerant eastern oysters. So a little bit of background of why this research was done. There's been a lot of change in our coastal areas, mostly due to climate change and human influences. So with climate change, we see a lot of rising in sea levels and heavy storms. With human influences, we see a lot of loss of infrastructure. So as climate change happens and human influences happen, we'll see a lot more erosion of our shorelines. This research was funded by the Department of Homeland Security, and they have a coastal resiliency program. If you've ever seen like the maps where it's like these places will be underwater in like X amount of years, that displaces large populations of people. So that's why it's been an issue lately with DHS. So the importance of oysters in living shorelines, they remove excess nutrients such as nitrogen and phosphorus very successfully. Reefs remove 7 times more nutrients than watersheds that don't have successful oyster reefs. They also create habitats for crabs and other fish, help them hide from predators. They support the economy and seafood industry with local and state governments, and just overall improve the appearance of the bay and make it look better. So a challenge with this research is the salinity gradient of the Chesapeake Bay. If you look at the little star right there, that's us. We're in the mesohaline region of the bay, but there's a huge gradient. Down at the bottom is the highest it opens up into the ocean. But as you move in, there's more fresh water mixing in. So the salinity goes down almost as low as 0 parts per thousand up in the northern parts and the rivers. But you can find eastern oysters all throughout the bay. So they're all throughout the salinity gradient. Melting snow in the spring months does cause a decrease in salinity, but then as the weather dries out, it gets a lot higher. So it changes throughout the year as well. So a little bit about oyster larvae. I know for me, before I started this research, they're just kind of like an animal that existed. I didn't know what like the beginning stages were. So this research focused a lot on the larval stages. They start out as the fertilized egg, and then they go through the 4 stages of the larval process. They range between like 60 to 300 micrometers, so they're really not very big. And I just wanted to highlight some structures that we focused on. So the cilia, when they're in the beginning larval stages, it helps them swim around and eat. And then as they grow, they get to the pedivelager stage, which is pedi for foot, and they get the eye spot, which helps them orient, and the foot that helps them settle and set on the substrate. The gut, which is what holds like the algae and helps to digest it, helped us a lot as well, because that was a big indication of health. If it was darker, then the culture was healthier. So the first stage of this experiment was the larval survival. We had the 2 lines, the low salinity tolerant and the Maryland wild type, and we wanted to see the difference between artificial seawater at about 14 parts per thousand, and then ambient. We call it ambient because it kind of changed throughout the experiment. It was taken from the cove out here, so it's usually 9.5 to 12. So first we spawned our Maryland wild type and low salinity tolerant oysters. We used strip spawning, which is surgically removing the gametes, and we combined the eggs and the sperm, which allows for more controlled fertilization. And then this was our experimental design. So we had the replicates for both lines and the 14 and the ambient salinity levels. 3 times a week, we would change out the water, we would collect the larvae and measure the survival and growth. That's what they look like under the microscope. Oops. And then this was our data sheet, so we were able to look at the health and track that, as well as get counts and average out the population and size. So this was the results of our larval survival experiment. You can see there's really no steady increase or decrease between any of the lines throughout the experiment. It was highly variable, and it was decided that it wasn't significant to decide whether there was a difference between the 2 conditions. This is the larval growth results. Once again, there was no steady increase, or it should be an increase because they're growing throughout the experiment, but it was highly variable. So it was also decided as not significant for the data collection. The second phase of this experiment was the settlement experiment, where we wanted to see what the settlement rate of the two lines are in the ambient and high salinity conditions. It was a pretty similar setup. We didn't use our larvae that we spawned because there's a lot of issues with the survival. So we kind of wanted to start fresh. So we took the low salinity tolerant larvae from Horn Point Hatchery, and then the Maryland wild type from Piney Point. It was the same with the replicates and the salinity. We just put oyster shells in the bottom of each bucket to allow for settlement. So this is the data collection. Although the means for the low salinity tolerant line were higher 2 weeks post settlement, there was no significance between the two settlement rates. And then this is 1 month post settlement. Once again, the means were slightly higher, but no significant difference between them. And then this is a visual. So 7 days post settlement, they're really tiny, but you can see them on the shells. You can mostly see the dark guts on the oyster shell. And then they grow pretty quickly throughout the experiment. You can start to see color. They get quite a bit bigger. So one of the biggest issues for this experiment was our water quality. There was a lot of contaminants such as ciliates, rotifers, nematodes, other types of algae that we didn't put in personally. And these will outcompete the larvae for space and food. They're usually quite a bit bigger than the larvae. So the bigger organism is going to get food and space, and it's very detrimental to the health and growth of our larvae. We also had clustering during the settlement experiment. So you'll see higher settlement rates in the beginning of the experiment, but as they grow, they're going to outcompete each other for space and food. So when they're all clustered together like that, they start to grow into each other and will have less spat making it to adulthood, which can be a major problem later on. So this was the low salinity tolerant line for the spat counts. The averages were about in the range from like 20 to 50, but the yellow highlighted were spat counts that were high above the average, so that's a clustering that you see. And then the pink was averages that are a good bit below the average for spat counts. And then that was the Maryland wild type. So to conclude this data, the low salinity tolerant line and the Maryland wild type line showed fairly similar survival, growth, and settlement throughout the experiment, but it was highly variable within each culture, so we were not able to conclude any significance with the data. One thing is that our salinities were fairly similar to each other. 10 and 14 is not that far off. So if we were to do future research, we could do a more drastic difference between the salinities. So like 5 and 20, we might see more differences. Contaminants was likely the cause of the variability. It can be very detrimental to the health. But overall, the best method that we believe in looking at past research is taking native oysters from that area that will be acclimated to that area of the salinity gradient and using that to repopulate and restore the oyster reefs. So I'd just like to thank my mentors, Dr. Fan, Tamika, and Brittany and Amanda, as well as the fellow interns and the Pearl staff. And then any questions?
Back to your seat. On Zoom yet? You explained it so well. No questions for you.
Okay, I'll kick it off. Let some questions in interim, but maybe I missed this, but so you use the 14 parts per thousand for the artificial seawater. You're exploring, one of the things you're exploring is how well the low salinity tolerant line does, compared to the other line.
Right.
I'm curious why you might not have used like a lower level of salinity to, isn't that ultimately a big part of the point, is to see if this low salinity tolerant oysters can survive at like, problematic levels of salinity, like 5 or 8 or something? Like what's the thought process behind that?
So the 14 is more of like a control. We know that Eastern oysters usually prefer 15 to 20, so 14 might be even a little low, but since we did use oysters native to this area, the ambient, which was supposed to be the low salinity, ranged from about 9.5 to 12. So that would be great for future research. If we did 5, that's a really low salinity. See if there is a difference between the low salinity tolerant and the native oysters.
Thank you.
We have a question from Zoom. Are there any environmentally healthy ways to reduce or eliminate potential parasites who pose risks to the growth process? It's definitely tough since they are so small to filter them out of the water. Just, I guess, getting a higher quality of water filtration would help. But obviously that's expensive and not always attainable, so. Okay. So we're gonna kick it back off with Aaron. So we've got 3 more presentations, then we'll give you a little wrap up spiel, and then we've got food for you. So I'll let Caitlin introduce Aaron.
Great. Hi, everyone. Very excited to introduce Aaron Jackson. He's the intern working on this project with the econ team. So he's been working with myself and Dr. Scott Kanaki. Give it up for Aaron.
Hello, everyone. My name is Aaron Jackson. I'm a recent graduate here at Frostwick State University. I study geography, and my project is the levels of trash impacting outdoor recreation. So just some background knowledge about the Anacostia River. Is located in Washington, D.C., along with other regions within Maryland. The Metro Maryland region, through the EPA, also known as the Environmental Protection Agency, it has been deemed to be a highly polluted river, which is listed as a 303D. So for those who are not familiar with a 303D is, it is a list of impaired streams that are basically due to high levels of pollutants. The Environmental Protection Agency, Maryland Department of Environment, and the DC of Department of Energy and Environment have been taking the time to revise the TMDLs. Again, so for those who are not familiar with TMDLs they are total maximum daily loads, and they basically establish the maximum amount of pollutant within a water body. So the Pearl team, us, we're going to be conducting research and trying to also revise these TMDLs. So we're going to create a survey. So briefly, this survey is going to be sent out to the general population. It's going to touch areas in the Maryland, DC, and some parts of Virginia. And basically, the visualizations provided will give everyone a human perception to determine what levels are more so unacceptable or acceptable. Previous literature. So again, what has been explored has been pollutants impacting water-based or adjacent research. So the 2 main studies that I've been studying have been JICUS and the Supley paper. So again, this approach that they have been studying, this is like the first study. So they are studying different algae levels within water streams. So determining how it's impacting outdoor recreation. So we're kind of using this approach and kind of making it more so unique because it's never been done with trash before. Also, just to touch bases on our curling approach. Again, this focus on debris, on how it affects in the marine environment. As you can see, here is a screenshot of our potential stream segments. So again, these are a total of 7. And what we do is we evaluate each one, and for our project that we best suitable, we will be selecting 2 that meet the requirements for our project. Here's our stream evaluation rubric. So this is how we grade the stream. So we take the time to go out to each stream. And again, the main things that we kind of look for is accessibility. So once we get there, how long does it take for us to reach the stream? So sometimes it can be anywhere from just getting out to our vehicles and heading straight to the stream, or we have to walk a few distances. Ideally, we want our stream path to be at least 100-foot straight stream path segment, along with making sure that it is safe when entering and exiting. There have been a few stream sites that have been kind of more so non-shallow and high levels of water, which make it more difficult and kind of be unsafe to not knowing how you enter and what are you stepping on. And again, lastly, to support the streams within the rubric, we want to make sure that it's able to have several types of water recreation activities. Here is a ranking potential of our stream sites. So again, there are 7 in total. The max you can score is a 21. So the 2 highest are Northeast Branch and Slago Creek. Overview of the streams, Sligo Creek and Northeast Branch. So the top picture is the picture of Sligo Creek. It is located in Montgomery County, Maryland, and the length distance is 9.1 miles. Personally, these are the two streams that we'll be working with, and the first one is my favorite. This area is very peaceful and is more so secluded. And then with Northeast Branch, it is located in Prince George's County, Maryland, with a length distance of 3.2 miles. I'm describing Northeast Branch. I would say it's more so of an enforced embankment. So this gives us two different stream styles and how like they are different they're formed differently. And so this can show us this can allow us to look at the different types of streams and how, even though they're different, they have some similarities. Here is a trash load scenario pie chart. So again, these 10 objects listed are our trash categories. So for example, plastic bags, carryout bags. So this data has been collected through COG. So for those who are not familiar with COG, it is DC Council of Government. And again, so the percentage underneath is a representation of a 5-year average trash distribution within the Anacostia River. So when we do our trash load scenarios, ideally the number within the pie chart remains the same. The only numbers that are changing is the amount of trash and then the weight by pound. Also, again, like how I mentioned that we do keep the percentage the same, it is important to maintain this control, because without this control, it would make the studies kind of a little messed up. But again, previous studies have been shown that people do perceive different types of trash differently. Here are some images taken. So on the first picture is Caitlin and Emily. So we'll just take some nice pictures. And then the other two pictures are just an idea of this type of trash that we collect. So there's a whole bunch of trash. I would list it, but I'll do that later. Our first approach, exit photos. So exit is moved from its original place. So what outside of the environment, so its current environment. So the exit stage photo approach has been shown in previous study. Also, what I mentioned, the curling. So our first picture will be an example of our 0 pound scenario. So within our 0 pound scenario, there should be no trash visible. And in our last photo on the right, it's a lot of trash. This will be our 200,000 pound scenario. So again, ideally, when taking these photos, it is important to make sure that I keep the same angle and to make sure no trash is overlapping nor shadow present within the photo. Our next approach, in situ photos. So what is in situ? So it's kind of basically the opposite. So it's within its original place, inside the environment. So this display allows us to look at the visualizations. So these pictures presented to you now, these have been practiced here at the Pro Pier. So ideally, again, making sure I keep the same angle and being able to make sure this trash kind of looks as realistic as possible. When the survey is once the survey is deployed and people are looking at these visualizations, we want the public to look at these photos and not just say that they were just placed there. We want to look as realistic as possible. Here is a quick video of Northeast Branch and just the side of the embankment. And this is how it would look for a giraffe run within our survey. Challenges and solutions. Okay, so when I first came onto this internship, I would just say, like, again, like when you're working with any project, of course, you're gonna have a lot of obstacles and hiccups. But one thing how I like about the economics team, I like how we can just all come together and just meet and we can just talk about the challenges and just be able to provide solutions and just get feedback on our ideas. So using the GoPro stabilizer and our camera handheld. So when we're entering the streams and trying to handheld it, and then obviously just trying to move accordingly, it can cause a little bit more stabilization. So interns, you guys want to show it off? So this is the mechanism that we designed. It's a boogie board attached with a GoPro. So this design helps us create, so the boogie board, it helps us helps it float in the water along with the GoPro stabilizer. So the technology within the GoPro has a built-in stabilizer that helps the videos and pictures be more stable and stabilized. Challenge number 2, the X2 approach. So again, the pictures that I showed previously in my PowerPoint, having those pictures on the white tarp. So ideally, like when taking it, again, making sure the picture are keeping the same angle. And I had to actually stand on a tall ladder to make sure that the lens is correct and proportional. Within the GoPro, even though it is a great model for recording, but when it comes to taking pictures, the wide lens is very long, so you kind of get a lot of it. And I know there are a lot of technology to crop many, being able to crop most of the stuff out the picture, it wasn't really ideally. So the solution for this problem was using an iPhone camera and just using the correct lens. It was actually 0.5 meters. Challenge number 3. Scientific research permit requirement. So ideally, so when we were doing this project, we thought, we could just be able to just throw trash in the water and just, take pictures of it. So, obviously you can't do that because you go to jail for littering. So we had to contact the park, and they're going to request us a permit. So the next time we go, there'll be a park ranger with us. So they'll be able to just monitor us and making sure we'll be able to take these pictures, and so we can create the survey. And then challenge number 4, software editing issues. So Adobe Express. So the video I showed you guys, it's an okay video, but as you can see, there was a little bit of kind of like too much movement. It wasn't as stable. However, like since I have the GoPro, if anyone's interested, I can show you the video. But when you guys are actually looking at the GoPro video, it looks very stable, but like when you blow it up on Adobe Express, it's not. So we're just gonna take some steps back from the video and focus more so on the visualizations. Further research. So again, since my internship is coming to an end, I'm still going to communicate with the economics team and still check on, because this is an amazing opportunity for me to understand how this affects the environment and how this can hold so much value. Again, the people within the economics group will continue to work with the work groups and meet with them on a daily basis and continue to show their progress and get any feedback that can be relevant towards the study. And in the ideal goal, since this is a 3-year project, the survey will need to be deployed within early 2023. I just want to give a big shout out to my family. Unfortunately, my family could not make it. Friends, guests, and especially the Morgan State Pro team for accepting me as an intern and just allowing me to be me and giving me this amazing opportunity. Any questions?
Questions for Aaron?
What is the most interesting contrast and
Repeat the question, please.
Say it again.
Repeat the question.
So the question was, what was the most interesting piece found here working through the Pearl? I would say we collected a Dora the Explorer blanket. It was like really long and very dirty, but it was kind of cool. Yes.
Is there any comparison of Rock Creek and the condition of the water now as opposed to say 30 or 40 or 50 years ago? The reason I ask is I used to play in Rock Creek as a kid, which was 50 or more years ago. We didn't think too much about it. We didn't drink the water. We'd go down there and play it. And I also have wonderful memories in the winter time with friends. We'd go down there with a fresh beer can and sticks and play hockey. It was nice to see that we're still working on it. But I was wondering if it's degraded or about the same, or if there's any way of knowing.
I would say from my perspective, I would probably say that it's the same.
Okay. Wonderful.
Thank you.
Thank you, Aaron.
Oh, yes, okay.
Yeah.
Okay, so it is my great pleasure to introduce Katie Dolph, a rising Morgan senior who has been with the Morgan Pearl team for the past 2 years. So she's had, I think, a pretty unique experience working with myself and Caitlin Ritchie and Abu Bakar and others on this project. She was in it from the get-go on the ground floor, helping us develop a survey of cicadere hunters. And we did the survey while she was taking classes this past year and had a great response rate. And so Katie, we're grateful that she came back to the Pearl to help with data analysis, report writing, and doing some pretty dang interesting and innovative analyses that she's going to show you here. So without further ado, Katie Dolph.
Hello, everyone. Thank you, Dr. Kanaki, for that introduction. As you mentioned, I'm Katie Dolph. I'm a rising senior at Morgan State University, majoring in biology. And this is my second summer at the Pearl working on the cicadere project with the econ team, working under Dr. Kanaki and Caitlin Ritchie. So it's assessing hunter opinion and economic aspects of cicadere hunting and management on Maryland's Eastern Shore. So before I get into our research, I just wanted to give a little context and in the importance of recreational hunting at large in Maryland. National surveys do a 5-year approach to gauge participation and expenditures for hunters across the country. So specifically in Maryland, they estimated that 88,000 hunters had spent 1.4 million days hunting in 2011. They had spent over $200 million in these hunted-related expenditures. Of that, over 100 of that million was directed directly into trip-related expenditures, and this generated over $400 million in regional economic impacts that supported over 4,000 jobs, and that generated over $100 million to support those salaries, wages, and business incomes. So from this data, we can see that regional or recreational hunting can have these regional economic impacts that support economies, but national surveys like this at large are broad or too broad to have the scope of research of cicadere, which is our scope of interest. So a little bit introduction into our cicadere. Although being named cicadere is actually a member of the elk family, and it is non-native to North America. It is native to southern Japan, specifically Yukushima Island. And so they were introduced to this area over 100 years ago in the early 1900s when 5 or 6 individuals were released on James Island, which was right off the coast of Dorchester County on Maryland's Eastern Shore. So they prefer the marsh and wetlands areas compared to our native white-tailed deer, which stays in more of the forested canopied areas. So in the 100 years that it's been established in the Eastern Shore, it's become limited to the Delmarva Peninsula and Maryland's Eastern Shore, because this is one of the only areas in the US that you can free-rangely hunt this species. And so they create this unique recreational hunting experience that has been gaining recent recognition by being featured on Netflix series, certain podcasts, and multiple social media platforms. But despite this, only biological and ecological studies have been published in the literature. So we want to fill those gaps by researching the economic and human dimension studies on the cicadere hunting experience. So with the research and data that we got from our survey and Maryland's DNR's Hunter Maryland survey, we are able to estimate there are 5,425 cicadere hunters, both resident and non-resident to Maryland, that had spent over 63,000 days hunting of effort in the 2021-2022 hunting season. And the Maryland Department of Natural Resources was able to estimate that there are 15,000 cicadere in 2018, and this is the largest free-ranging cicadere population in the United States. And so with our data, we were able to estimate that 89% of the hunter's effort has been targeted in Dorchester County, their effort in harvest. And the landscape of this county is a little bit more rural and economically challenged. They have the 4th lowest median household income and the 3rd highest poverty rate in all counties in Maryland. So we want to examine if these recreational hunting experiences can benefit their economy. So that brings us to our project goals and objectives. So we wanted to develop and deploy a hunter survey to gather data on their expenditures and how hunters, they form their opinions on the current regulations on cicadere.
And so with that data, we want to be able to estimate the total annual expenditures the cicadea are incurring and better understand their current opinions on the management of this non-native species, and eventually develop a report for the Maryland DNR with our key findings and any management implications that we can recommend for this non-native species. So it brings me to our survey methods. So we launched our hunter survey at the conclusion of the 2021-2022 hunting season in February. So we developed this survey instrument with the collaboration and funding of the Maryland DNR. We had pre-tested the survey with 10 Maryland hunters to make any revisions to the final draft. Our sample frame was over 96,000 resident and non-resident hunters in Maryland. And as you can see here, this is what they would receive in their email, inviting them to participate in the survey. So it was an online web-based survey that was emailed to hunters that had purchased a license in the past 3 years and that had provided an email to the DNR during the registration process. And so they had 2 forms of contact soliciting participation into our survey. So the initial email blast and 1 email follow-up 1 week after that initial invitation. So our response rate, as Scott mentioned, we did get a big response rate of over 3,600 viable responses. Unfortunately, we were unable to calculate a formal response rate because there is no specific cicadea hunting permit. So our hunting sample was all hunters that had provided an email and not just cicadea hunters. And we were able to gauge cicadea hunters from the first question of asking if they had ever hunted a cicadea in Maryland. And if they answered no to that question, they were directed out of the survey. So another integral part of the methods this summer was using Stata, which is a statistical software program. So prior to coming into this internship, I had no experience with any software program. So it was a great experience learning how to write codes and commands to manipulate and visualize the data. That was a key part in our data analysis portion and cleaning up our data set to make it tight and clean. So after our data set was clean and ready for the data analysis portion, we were able to get data from our questions in the survey. These are just screenshots from those questions upon regulation and their estimated trip expenditures. So in the next following slides, I will be examining some of our major conclusions that we received about hunter opinions on regulations, such as antler point restrictions and bag limits, and also explaining our estimations of their total annual spending, the number of public and private hunters, and the amount of trips that they took during the 2021-2022 hunting season. So beginning with antler point restrictions, our survey was able to gauge this by a Likert scale question upon their acceptance and non-acceptance of the current regulations. So currently the regulation on antler point restrictions, there is no minimum antler point to harvest a stag, which a stag is an antlered cicadea. So we were able to get data upon 44% of hunters found the status quo to be acceptable upon the status quo, which is the current regulation for cicadea. But interestingly enough, when we examined and brought into the equation the alternate 1, which is a minimum point or a minimum of 2 points to be able to harvest a stag, increase of 52% of hunters found this to be acceptable. So it's an interesting observation that hunters are more accepting to a change in this regulation. But that regulation would only tolerate to a certain amount, because when we had a third alternative to a 3-point minimum to harvest a stag, a whopping 83% of hunters found this to be unacceptable in a change of regulation. And in terms of bag limit, we had a similar approach to this Likert scale acceptance. So the current status quo for the harvest and bag limit of stags is you can harvest 3 stags in the total season upon all 3 weapon seasons. So we found that 49% of hunters found the current status quo to be acceptable, but also interestingly, similar to the antler point restrictions, we found that a 10% increase to 59% of hunters accepted a change in bag limit to a decrease of 2 stags to be harvested in the given hunting season. So this is an interesting opportunity for wildlife managers to be able to make a change for the better and enhance the hunter's experience upon regulations. So secondly, of our results, we were able to estimate annual trip expenditures. So we were able to estimate that hunters spent 11.6 million in total annual spending. 7.7 of this was private land hunters and 3.9 million was public land hunters. We also explored a 95% trimmed mean, which reduced this total spending by about 15%.
In our survey, we were also able to ask about the allocation of this spending, and the majority of hunters indicated that 75% or more of their expenditures occurred in the county that they hunted in. We also able to estimate that about 2,800 public land hunters took over 10,000 trips, and 3,400 private land hunters took over 15,000 trips. So a little bit of discussion from our results. So we were able to understand that hunters are accepting of new regulations compared to the status quo. So this is an opportunity for the wildlife hunters to make a change in the regulation to again enhance the hunter's experience and also the ecological stability of this non-native species. We are also able to understand that of this over $10 million that they're spending in trip expenditures, most of that is staying in these counties. So it has potential for economic significance for these counties that are a little bit more financially disadvantaged and to support their economy and local businesses. And like I had mentioned, is one of the only places in the US that you can hunt this animal free range. So it is a source of tourism that also brings in new revenue for the economies on the Eastern Shore. We're also able to understand that private land and archery had the highest expenditures. So private land, usually a lot of outfitters and guide services use private land, so they charge a fee. So that is a driving force that increases these expenditures. And also archery season is by far the longest season. So hunters are spending more days and spending more money in their effort in archery season. So with our research, we were able to understand that stick deer hunting does have economic significance that can support these economies, and we can implement or provide recommendations for the wildlife managers upon regulation. So I just want to thank the Pearl for hosting this internship, specifically my mentors, Dr. Scott Kanagi and Caitlin Ritchie, for all their guidance and support over the past 2 summers. Dr. Knobloch, the coordinator of our internship, for making this all possible. I want to thank the Maryland DNR for their funding, and specifically their game mammal section leader, Brian Eiler, for help with the data. The Chesapeake Research Consortium and Randy Rao for hosting me as a C-Stream intern. And last but not least, the Cicadere, because without them, we would have no research project for me to work on. So I just want to give a big thanks and shout out, and also to the fellow interns for all their support this summer. Yes, sir. Oh.
Are there any considerations of changing the regulations on the secondary limits or bagos or emphasize?
Well, currently we're in the stages of developing the report with our findings to submit to the DNR. So that's upon their review and their discretion to change the regulations. But once we get that data, I'm sure that there'll be some talk of revising any regulations or talk of regulation revision. Yeah.
Sure. Why do you think that hunters were more in favor of a decrease?
I would think that the hunters that are more in favor of that are thinking more long-term about the future of this hunting experience, especially with the antler point restrictions, that maybe different hunters have different motivations. And if they're more of a trophy hunter and they want stags with larger antlers, they would want the young stags to be able to develop and not all the other hunters be able to harvest those deer. So giving a chance for them to develop and future opportunities for hunting. Yes, sir.
As a rising senior, how might you see the new acquired skills in programming and data analysis work for you in the future?
Yeah, I definitely had a struggle learning data, and it was an awesome experience to be able to learn that. Like I said, I had no prior knowledge, so I think it'll be a great tool that I can bring back to Morgan State and apply to any of my upcoming studies or different classes I could take to implement my statistical analysis programming skills.
Got 3 Zoom questions. Do you feel it would feel it more hunters to have tighter, more solid regulations, or to loosen up these regulations and leaving more other?
Well, upon our data and estimating these changes in regulations, it does seem that hunters have a stronger acceptance for more stricter regulations. And as I mentioned, it could be, you know, a future generational thing to sustain this species. So I think that hunters are receptive to changes in regulation.
Given that second year are non-native, what are the goals of DNR? Do they want to maintain or eliminate the population?
Upon research, they want to maintain its cultural carrying capacity so that it's not harming the native species here or the native environment. And so their populations aren't exploding, but they don't want to necessarily eradicate this non-native species.
So local governments and even at the national level must have serious education and public service programs and announcements that seriously address the tragic problem. This is actually. It will take time to think. All right. So that is all of our questions for Katie, I believe.
Yes, sir.
Has there been any observation of a decline or increase in cedar population in the last, say, 20, 30 years? I've been hunting them for about 40 years and I have 85 acres of land in South Georgia. So I'm intimately familiar with the cedar and the hunt. So I'm quite interested in this.
Yeah, well, the last estimation that I mentioned is in 2018, 15,000 individuals. And I think that the 100 years that it's been here, their population has been steadily increasing. So I'm not sure what the current data is on their current population, and that's just an estimate, but I do think that their population is growing. Yes.
Yeah, can you go back to expansion slide? It was hard to hear that grass cutting activity, but I hear you did something looking at 95%. What is there?
Yeah, so we also explored a 95% trimmed mean. So that was accounting for any outliers. So survey respondents that had either extremely high or extremely low estimates. So this just gave us some more conservative estimate of total expenditure. So that reduced it by about 15%.
Thank you.
Oops.
I didn't just have one project, she had to have two.
I'd like to introduce Jessica again. She was an overachiever, as Amanda said, and did 2 projects this summer. So excited for you guys to hear about what she did with soft shell clams. So come on up.
All right, I'll wrap this up and keep it a little bit shorter because we all know it's time for food soon after this. And all right, so this one's gonna be a fun one. So pushing the limits, heat tolerance in soft shell clams. So first, what is a soft shell clam? It is the image on your left. So that is a soft shell clam, also known as a Maya arenaria. And the common names are soft shell clams, and/or steamers. So it is found in the red area on the graph to your right from Maine to Virginia and has some of the highest concentrations in those points. So the economic value in Maine alone, it is a $5 to $10 million industry annually. And in 2019, the industry was valued at $54 million with wholesalers, distributors, and others. Okay, so the history in Maryland, they are native species to Maryland. So they were very popular in the 50s to 90s. As you can see, the blue line on this graph, that is the number of bushels that they actually caught during this time. So it was a very popular species to catch, but it started to decline due to overharvesting and other factors like that. And even though the value skyrocketed in the 90s, it is almost non-existent today. So some of the main species in the Chesapeake Bay currently that are commercially for the aquaculture industry are oysters, blue crabs, Atlantic menhaden, and other ones. So I'm going to focus specifically on oysters. So this graph to your right is the number of bushels of oysters in the Chesapeake Bay from the 1960s to 2006. And as you can see, the population size is decreasing. Like in Maya's presentation earlier, she talked about how global warming has been affecting the populations and causing a bigger spread of disease. So other factors like overharvesting, disease, and pollution are decreasing these populations. And so soft shell clams creates an opportunity that it creates a new species for aquaculture. So in case the oysters do bad this year, they have something else to fall back on. And furthermore, the softshell clams are more tolerant and grow faster in low salinity conditions than oysters, which in Kayla's presentation, she mentioned all of the low salinity environments near here and how that can change very rapidly. And it reaches commercial size in 2 years or less. So some of the challenges for developing the softshell clam culture in Maryland is seed production. So what happens normally for aquaculture industries is that they get the seed from hatcheries like the one we have in our actual Pearl laboratory. But softshell clams, because this industry has not been big in a while, there are no seeds to do it. So they would have to go out and physically get their own clams by themselves, which would be hard to start out. And second, the culturing method, because usually in places like Maine, they have large stretches of land where you can actually put the softshell clams in these intertidal zones. We do not have that opportunity, so we would have to start something from the ground up and actually develop something for our region. And third is the heat stress conditions. So they usually survive from -2 degrees Celsius to 28, but they prefer from 17 degrees Celsius to 23. However, in the Chesapeake Bay, they usually do not survive over 28. This creates a problem because just yesterday, it was over 29 degrees Celsius right in our pier. So it's definitely trying to find a species that can handle these waters. So our objectives are to achieve successful breeding, improve the heat tolerance, and selective breed based on phenotypic and genotypic breeding methods for heat tolerance. We've actually already achieved the first one. So Dr. Lou and Brittany Bryant were able to successfully spawn juvenile clams in our laboratory. And we now have around 468,000 juvenile clams. So we're just focusing on the other ones to get better. So the two approaches that we're thinking of using are phenotypic versus genotypic. Phenotypic would be we heat shock a large number of them and then take the survivors and breed that way. Genotypic is finding the specific genes that deal with heat tolerance to then use in better studies and to find the exact clams that have this. So my original project was going to be on the phenotypic approach, where we would heat shock around 600 adult and juveniles. However, this is the pier conditions and the temperatures of our pier right off of Pearl. And the blue line is when we actually took out the clams, and above the red line is above where they are comfortable. So that circle is the amount where the pier conditions spiked over this. So this has resulted in a huge mortality. So that left around 75 adults and 150 juveniles. So this was not an option at this time. However, we could still run the genetic approach to find those genes. So we still heat shock a smaller amount of these clams and sample the alive clams to get those genes. We then will send it to a lab to have their RNA sequence. And through these processes, we would identify the heat shock genes. We would identify the markers. And then this would help us specifically find the clams that we would like to breed later on. So what I did, we did a smaller heat shock thing. So the tank on the left is the control temperatures from 19 to 23 degrees Celsius. So these are the ones mimicking their ideal temperatures. And we have the tank on the right, which is heat stress, which was over 30 degrees Celsius, which would normally mean that this would kill almost all of them. So as you can see, there are also two different types of methods to actually hold them. So as you can see here, this is called a clam bottle, where we would place one clam in a bottle filled with sand. And then here are clam sandwiches, which I will show better here. So we also wanted to see which type of thing to hold them was better, because we don't have a specific method to develop these the best. So we're trying to find out which holding one would be better for it. So some pros for using the bottle would be that the sand could insulate and protect from challenging conditions by mimicking the natural environment. And it prevents from overcrowding because there's one clam per one bottle. So, and also it helps protect against accidents, because if you're going to be harvesting them, you need a large one. And if you accidentally drop them because you're dealing with hundreds of them, it would be better to have some insulation. For the sandwiches, the pros are they're so much easier to maintain and set up. You can have a higher density, there's a lot less prep work, and you don't have to deal with the sand. So some of the observations, the controls are on the left. It's a little hard to see in this light, but one of the things for heat stress, like right here, that is a siphon extended. So that's the filtering organ of these creatures. And here they're kind of more buried in the sand. So in the heat stress, more of the siphons were actually extended, but here you can see in the control, it's a little more open. So it's filtering better than the heat shock, where their siphons are a little more closed. Furthermore, their siphons responded slower to prodding when they were in heat stress. So they were very challenged by these conditions. Okay, so here is the percent mortality in the soft shell clams between the bottles and the sandwiches. We wanted to see which one was better. So in day 3, the sandwich actually had the first mortality and it had a higher mortality rate consistently over all of our days. So they almost had 90+% that died in the sandwiches compared to the 60-ish% in the bottles. So our next step was, this was the genotypic approach I mentioned earlier. So we actually took the samples from the live clams. So the first death was measured on the 27th of July. And so we sampled from 3 clams from each group. So 3 from the sandwiches, 3 from the bottles from each tank. And so we took samples from 7 of the organs. So their gills, their heart, adductor muscle, foot, mantle, gonad, and siphon. We took those different samples to measure the different upregulation or downregulation of the heat shock genes in those different organs and see which ones we need to focus on for later research. And we also took another gill sample to actually have it be RNA sequence. So here is me and Dr. Liu going and actually taking the samples, and we processed the RNA in that lab that you saw Maya was in earlier. And so we got the RNA prepared for sending off to the lab. So in conclusion, in heat stress, the more clams in the sandwiches died. However, in the control, no clams died, so both methods work. So in the challenging conditions, the bottles might be a better alternative, but if you're going on a large scale and don't have challenging conditions, I would personally do the sandwiches because they were a lot easier to maintain and a lot more practical. So our future steps would be those samples you saw us take, send them to the lab, get the candidate genes back and validate them. And then we would use those genes to actually breed and pick some of the top clams that we have that have these genes, and then use this data to spawn the best individual to run trials on those offspring for later. We could also continue the bottle versus sandwich research to have a better method for other aquaculture industries. All right, and I would like to thank the Maryland Sea Grant and Morgan State University OTT IGAP grant. All right, here are my references. And are there any questions?
Go ahead and do the Zoom comment / question. Can you make clam sandwiches on rye? Just kidding. If current climate change trends continue, how feasible would it be super clams that breed super clams who can withstand and thrive at even higher temperatures?
So if we can get as much, we will keep testing on the heat tolerance genes. If we are able to find the heat tolerance gene and actually manipulate it in the way we want, potentially we could go as high as we can for the changing conditions happening. Okay, I'll end it. Wait, can you go to the next slide, please? Also. Yeah. Okay.
Should be able to.
So I'm going to graduate school soon. If anyone knows any programs or anything like that, I'm gonna major in biological sciences. I am interested in aquaculture and anything related to biology. So please, here is my email. You may take a picture and send me things.
All right, so that concludes our symposium. Thank you so much for attending. We've really enjoyed having these interns here this summer. I'm gonna be very sad when they leave because they've been such a great group. But otherwise, we thank you for attending. Our director, Dr. Scott Kanaki, is going to make a few closing remarks, and then I'm going to tell you how to get food.
I don't want to stand in between you and food, as Jessica alluded to, but I'm going to for just a couple moments. So first off, want to thank a lot of folks here. You know, thanks to the interns once again for wonderful effort throughout the year. And you know, our staff has really just totally stepped up and done an amazing job. And you know, first and foremost, Amanda Knobloch for just a wonderful job throughout supporting these interns, making their experience just hopefully something that's super memorable and transformative. So thanks, Amanda. So I want to thank you all, of course, too, for coming, our supporters, our funders, people that contribute in so many different ways to our laboratory. It really just means a tremendous amount to me and to all of us to come and share in this important moment with our internship program and learn about all the work that they've done throughout the year. So thank you for being a part of the Pearl team in so many ways. Want to share a couple of highlights about what's happening with the Pearl. Some of you know, some of you don't know. We've had probably 3 like monumental successes over the past 12 months. And you have to forgive me as director for thinking about dollars and cents often in these successes. But through a state of Maryland bill, the Pearl received or is receiving $2.5 million at a minimum and potentially up to $6 or $7 million to make an addition on our hatchery and really take the next step with our oyster and soft-shell clam aquaculture program. So that amount of money is a true game changer for our laboratory. It's something that we've never had that kind of investment. And I think that speaks to the quality of work that our staff is doing, that our students are doing, that the state of Maryland sees us as that type of being worth that type of investment. So that's number one. Number two is we've received a federal earmark to establish something we're calling the Semester by the Bay to take our engagement with Morgan State students to the next level. It's going to provide funding for 4 different projects that are going to heavily involve undergraduate and graduate students. Being 80 miles south of main campus is no easy task as far as engaging with students, but that's going to support a pretty awesome new program. So that's exciting. And thirdly, last October, Amanda Knobloch led the success ultimately before that, the development and implementation of a new degree program in coastal science and policy that is going to provide opportunities for undergraduate students to work at the Pearl, to study under Pearl researchers, and to diversify the marine sciences. So just want to give you some updates, some highlights. Thanks again for coming. Enjoy the food. I'll stop blabbing now, but thanks again, everybody. Yep.